Hi, everyone. My name is Daniel Gomez. This is for Biology 616 Journal Club for genome editing. The title of this is Retron Library Recombineering, or RLR, and it's going beyond CRISPR. I got this from an article saying, move over CRISPR, the Retrons are coming. This is a new gene editing technique that enables millions of genetic experiments to be performed simultaneously. So the idea of recombineering is efficiently creating mutations without breaking DNA. The methods are simple enough that they can be used in many cells at once to create a complex pool of mutations for researchers to study. Harvard Medical School created a new gene editing tool called RLR that makes this task easier. It creates millions of mutations simultaneously and barcodes the mutant cells so that the entire pool can be screened at once, enabling a massive amounts of data to be easily generated and analyzed. Some quotes from the authors. RLR enabled us to do something impossible to do with CRISPR. We randomly chopped up the bacterial genome, turned those genetic fragments into single-stranded DNA in situ, and used them to screen millions of sequences simultaneously. And that was Max Schubert, one of the co-first authors. RLR is simpler, more flexible gene editing tool that can be used for highly multiplex experiments, which eliminates the toxicity often observed with CRISPR and improves researchers' abilities to explore mutations at the genome level. And that was by George Church, and he was the senior author. So the idea behind Retrons is going from an enigma to an engineering tool. So there's segments of bacterial DNA that undergo reverse transcription to produce single-stranded DNA fragments. And these Retron exist as better known, they've been known for decades, but they didn't always know the function of the single-stranded DNA. So this kind of confounded scientists from the 1980s until June 2020, when a team finally understood and figured out that the Retron single-stranded DNA detects whether a virus has infected the cell. So this forms part of the bacterial immune system. Like CRISPR, it can be used for precise and flexible gene editing in bacteria, yeast, and even human cells. Another quote, we figured that Retron should give us the ability to produce single-stranded DNA within the cells we want to edit rather than trying to force them into the cell from the outside and without damaging the native DNA, which are both very compelling qualities. And that was Daniel Goodman, another co-first author. So RLR starts with a synthetic or natural DNA variation. It becomes a Retron plasmid library, and then the Retron Recombineering creates trackable edits. After that, you have selection and screening, and then targeted next-generation sequencing, so you can then see the phenotypic measurements. So the Retrons enable for a rapid production and screening of millions of trackable DNA variations and their effects on bacteria simultaneously. These libraries are either synthetic or natural DNA variants, and they can be incorporated into Retrons, performing the recombineering, and this produce mutant cells bearing the Retron plasmid that are available for targeted amplification sequencing using complementary primers to measure the mutant abundance in multiplex. So this is the paper that was published in PNAS. It's high-throughput functional variant screens via in vivo production of single-stranded DNA. And these are the cohorts. It's the Harvard Medical School Department of Genetics, the Weiss Institute, UCSF, Bioengineering, Gladstone, the University of Warwick, and MIT. Some of the sponsors were the U.S. Department of Energy, the National Defense Science and Engineering Graduate Fellowship Program, and the Space Consortium. The significance of the study is that this methodology reports a pooled construct of mutants bearing the precise genomic sequence variations and multiplex phenotypic characterization of these mutants using NGS. So unlike existing techniques depending on CRISPR-Cas directed genome breaks for genome editing, this strategy uses single-stranded DNA produced by the Retron element for recombineering. This enables libraries of millions of elements to be constructed and offers a relaxed design constraints, which permits the natural DNA or random variation to be used as inputs. Looking at the overview of Retron recombineering, we have the Retron MSR-MSD region, and that undergoes transcription. So you have the Retron transcript, and then reverse transcription happens through reverse transcriptase, a Retron reverse transcriptase that produces a multi-copy DNA or multi-copy satellite DNA or msDNA. So you have your 5' upstream variant and then downstream and then 3' in the multi-copy satellite DNA, recombineering donor. And then you have oligonucleotide recombinering. In this, you have synthetic oligos introduced into bacteria through electroporation. And if the bacteria anneals to the replicating DNA via the SSAP, or single-stranded annealing protein, and that anneals to the replicating DNA. And Retron recombinering, you have the Retron recombinering using the RT-derived DNA as a donor rather than transformed oligos. So this incorporates into the replicating DNA using the SSAP. Some of the results from the study. So this was a combination of several modifications to the genome and the Retron construct, which improved editing efficiency and demonstrated that the Retron sequence itself as a barcode to identify mutants within mixed pools, enabling the pool's screens of precisely created mutants. So this system allowed for simultaneously pooled characterization of precision mutations across the genome and offers advantages similar to CRISPR-Cas techniques. So this was the effect of endogenous factors on Retron recombinering. And then for the characterization and optimization of Retron recombinering, here you have the Retron recombinering plasmid, and it creates a multi-copy ssDNA or single-stranded DNA via the Retron reverse transcriptase. This DNA is then incorporated into the genome via the co-expressed SSAP. And then the editing is observed by the amplicon NGS facilitated by primers targeting the locus, which are in green. So you can see the primers in green and the locus in black, and you can see the measured edited fraction of the loci. In B, you have the optimization of RLR. So you have these colors differentiate GYRA and RPOB edits, and the biological replicants are indicated with the dots. So you have the two-sided unpaired parametric t-tests that were performed between the genotypes that are indicated with the brackets, and then the p-values are displayed as well. In C, you have the editing of cultures with GYRA editing plasmid expressing the red beta as an SSAP. So here you have, they edited the cultures in a turbidostat with continuous growth and induction of the mutant S and the SBCB and RecJ genotype that were used, and then transformed into the GYRA editing plasmid and expressing the red beta and single-stranded annealing protein. So these data, as shown alongside a simulated editing trajectory of an allele with a neutral fitness effect that showed an editing at 5% per generation. And then in D, you have the edited fraction observed across alleles after approximately 20 generations of induction and batch growth. So for that, you can see the Retron plasmid containing the CSP RecT as an SSAP that was expressed in mutant S, RecJ, SBCB in all cases. And the results of individual replicates are shown as dots, like previously. And then the results of the GYRA and the Repo B missense alleles are shown in B, are shown again in comparison for the TAG and TAA stop codon editing for 10 essential genes. So the mean edited fraction across there was achieved over the 12 loci were about 76.4%, and this is indicated by the dashed line here in D. And then in E, you show the effect of the Retron length on editing. So there are 6 replicates for each experiment that are indicated with dots colored by the locus. And then in F, you have the integration of a cluster of mismatch bases in the Retron donor. So you have editing on each position across 8 replicates that is shown for GYRA and RPOB alleles. So the observations have all mutations as reported as fully edited. And then a little bit more for optimization, so of exogenous RLR components. So here we have a population's genetics model, and it's describing the RLR process. For each generation, a cell has a probability R. So you can see the unedited cell, the probability R, being successfully edited by the Retron construct it bears. And then if it's edited, it propagates at rate F. So the F is determined by the fitness of this mutation in future generations. If it's not edited in a given generation, which occurs at a rate of 1 minus R, then the editing may occur at future generations, which is shown here and from green to orange. So this is a population genetic model describing the RLR process. So it's built, this is built they built a population genetics model that simulated the editing and growth of edited strains to explore the impact of editing efficiency on multiplexed experiments. So then the detection of phenotypes using RLR, the barcoded mutant pools, what happened here was a targeted sequence of Retron cassettes using that can be used for a measurement of mutant abundance in a population. Therefore, it's phenotype within a pooled assay. So here in A, we have the RPO mutations, including the RIF resistant alleles that were specified, as well as resistant alleles for unrelated drugs and control alleles expected to be neutral, lethal, or deleterious. And then in B, you have a pool of Retron plasmids conferring these alleles are transformed into cells, transformants are induced, and then edited, producing a pooled barcode mutant library. And then the selection is performed and the frequencies of the donors are compared before and after treatment of each allele. And then in C, you see the RLR enrichment values observed with the RIF treatment. And then the median of these replicates are indicated with a dot, and the error bars are the SE of the mean. So there are pseudo counts of 1 and given alleles not detected after each treatment, such that the frequencies are a lower limit of detection in these cases. So there's unfiled points indicated the alleles are not detected among any replicates after RIF treatment. And after filled points indicate alleles detected in a subset of replicates, there's an enrichment value of 0 that's marked by the horizontal dashed line. And this is for indicating identical relative abundance before and after selection. And then in D, you have the RPOB mutation, allelic enrichment. So that's across the concentration of the RIF that's displayed. So the median of these 3 independent experiments are indicated with a dot, and lines are connected as an allele across the concentrations of RIF. So antibiotic resistance is a growth phenotype that's clinically important. So they sought to first investigate the antibiotic resistance using RLR, and then constructed the Retron library confirming the known E. coli RIF resistant mutations, known M. tuberculosis RIF resistant mutations, and others, such as the neutral, the deleterious, lethal, and controlled mutations. So mutations observed in RIF resistance M. tuberculosis did not confirm detectable resistance in E. coli. So RLR enables high throughput pooled identification of antibiotic resistance alleles and facilitate the characterization of their relative effects across a range of conditions. And then the quantification, the quantification characterization of growth rate. So we have the pooled barcode mutant library, and then performing the screen as a time course, and then sequencing the Retron donors. So you have the NGS that does the sequencing. So in B, you have the relative abundance of donors during an experiment, and then the frequency of the alleles are normalized to the median of neutral control with each time point. And their strong abundance in the plasmid pool are shown there. So the measurements are within the gray rectangle, are occurring during construction, transformation, and induction of the library, whereas the white area occurs during the growth in the sub-inhibitory RIF, so the rifamycin. So the 5 micrograms per milliliter, and the mean of these replicates is indicated with a dot. And the error bars are standard error of the mean. So those horizontal dashed lines, a lot of people call those like thresholds, they're actually indicated to change there's no change in frequency. And then in C, you have the growth rate of all mutations in the pool that can be determined from the observed trajectories. So those are the RLR enrichment rate. And the growth rate was measured individually for 11 resistant mutations using the classical methods and plotted against the log 10 of RLR enrichment rate. So their error bars, there's some there that you can depict the standard error of the measurements, whereas R is the Pearson correlation and the coefficient between the two measurements. And then the P is the probability of the results, giving the null hypothesis of no correlation. So the degree of which resistant mutants outpace neutral controls is a quantitative measure of growth rate, which correlates well to the mutations when their growth is measured individually. So here in A, we have the RPOB mutants confirming the RIF resistance were constructed in the mut-S, rec-J, SBCB background via the oligonucleotide recombinering, and the growth rates were measured in batch growth with the sub-inhibitory RIF myosin. So the data shown was normalized to the parental wild type mutant S rec-J SBCB growth rate. And then in B, you have the summary of the RLR enrichment observed in a pool quantitative experiment over time. So here you have the RLR enrichment rate, which is the slope of all alleles for 3 replicate experiments that's indicated by the dots. And the different classes of mutations are separated by color. So RLR affords a quantitative growth metric for alleles within a pool that is comparable to tested mutants individually, but is inherently more scalable. And then after that, they did the RLR that detects causal variants using libraries prepared from evolved genomic DNA. So here we have the natural variation, the fragment DNA, the ligated adapters, construct Retron library, then selection, and then sequence Retrons, and then repeat the selection and then sequence again. So here we have genomic DNA as an input. So again, the ligated adapters are randomly fragmented DNA that enable the pool cloning, creating the multi-million member library of Retron plasmids. So the induction results in editing, and selection enriches for relevant mutations whose Retrons are sequenced as a pool via amplicon NGS. And then in B, we have the results of the genomic DNA screen. So these are deduplicated genomic coverage contained in the Retron library that's displayed, showing the mean coverage the mean coverage across 1 KB base pair windows. So these have been normalized to maximize the coverage. And then after the induction and selection of TMP, the coverage depth of variance is depicted for 2 replicates of the library, again normalized to maximize coverage. And that's shown in blue and light blue. And then in C, you have the full A locus that's displayed in dark gray arrow, and with the genomic position to the x-axis, and then sequence coverage observed for each base that's plotted on the y-axis as an input library. And 2 of the sequence observed were more than 1,000 times in the replicate A and are depicted as the pileup aligned to the genome and are colored by their abundance in post-selection sequencing. So detected SNPs are indicated by the vertical dashed lines, and you can see those at 49,765 CT, and then TG, and then another TG as well. And then in D, optionally, the mutant surviving selection can be transformed by the pool again. So screen for additional mutations and combinatorial efforts, transforming the library and to a strain that's already bearing the detected full A mutations, and that's full A star. So exposing the additional candidates variants that are adaptive and additional TMP, the z-scores for each allele describe their deviation from the mean allele coverage depth. So the variations with the z-scores over 2 have been labeled by the gene in which they occur. The coloring by their relationship is to their coding sequences. So this is directed evolution that can produce an antibiotic resistance, producing the mutant strains that are able to grow 1,000-fold more concentrated antibiotic than their ancestors. So RLR can be used to determine the causal mutations leading to resistance, such as isolates, by using random fragments of their genomic DNA to construct the RLR library. Most fragments containing a variant should be capable of editing, because we found that little effect of mutation position is correlated with the donor oligonucleotides. So the Retron Recombineering is flexible, just a generalizable tool for genome editing, which surpasses the editing frequency achieved by other markerless editing tools like oligonucleotide recombinering. And then pooled barcode mutant libraries can be prepared for a multiplex characterization of natural or synthetic allele variants. And this is the process of RLR. It enables millions of experiments to be performed simultaneously, observing the effect of mutations across the genome. It's an alternative to CRISPR-based methods, which also perform pooled phenotypic measurements of mutant sequences, but instead of using the guide donor method, it uses a donor-only method. So it eliminates the requirement for ablating the CRISPR targeting, allowing for the single base pair changes to be characterized without requiring additional the PAM or a protospace adjacent motif that overcomes the requirement for targeting a suitable PAM altogether. So whereas CRISPR uses the guide donor method, it actually decreases the performance as the distance to the PAM increases. So production of Retron ssDNA donor appears to improve CRISPR guide donor methods in S. cerevisiae and E. coli, and it's presumably marked by the recombination donor of abundance, and it's more abundant and accessible. So this lack of guides actually simplifies and streamlines the RLR method. So the sole requirement is unique, just a unique short donor sequence within the Retron. And this relaxed design constraint enables RLR using non-design variation. The lack of CRISPR activity may also enable use in systems for which CRISPR-Cas is toxic. So non-toxic editing methods like LR can be enabling new applications such that that were hampered by the expression of deleterious components, like the off-targeted effects. So the RLR is simple, it's streamlined nature, may lend itself to applications incorporating combinatorial sets of mutations, and strategies remain to be developed in this area. So here we saw that RLR measures beneficial growth and selective phenotypes, but non-growth phenotypes could be also made accessible with pooled measurements by fluorescent reporters, biosensors, single-cell transcriptomics, or a myriad of other screens. So RLR may find use as a method producing barcoded variation in constructs cloning into E. coli for other organisms, and it should not be limited to E. coli. The development of RLR can be used in other genetic systems, which is an exciting future area, which this work provides the roadmap. So there's a wide array of possibilities of future technologies using this flexible and simple editing and barcoding framework to accomplish the pooled mutant screens. Here's a list of materials and methods that they used. I'll just let you guys view that real quick, and then I'll go to the next, and that's pretty much it. Thank you for your time. You can reach out to me for any questions.